say that the last day is the way by which to measure the impact of the conference, because the room is not empty. It was an interesting conference, which he goes to show, which is great. So we're going to start with Thomas Höfer. We'll be talking about inferring lineage pathway dynamics and topology.
Okay, thanks very much. It's really been a great conference, and thank you for turning up at this early hour on the last day. I'll take up the theme, I think, that featured with some talks at the beginning, Becker and others at the conference, namely simply asking quite simple questions. Where do cells come from? Where do they go? How often do they do it? And in doing so, well, first let me get my acknowledgements across. It's really, most of the work I'll be showing is by Melania Barila and Jens Rösler. And Michael Floßdorf really played an instrumental role in starting off the whole ideas, and some of the scribble on the first slide was probably from Michael. It's a long time ago. And we're very fortunate to have a fantastic collaboration on, we have many, but in this field with Hans Reimer Rodewald, who's really an expert in making transgenic mice used for what's called fate mapping. And the data I'll show, and also the mice done by Katrin Busch, I will mention Weike Pai and Thorsten Feierabend in Hans Reimer's group. So I'll be concerned with how to make cells of the immune system in my talk. We're also interested in what immune cells do once they're made during adaptive immune responses, but I will just very briefly touch upon that work. And I was glad to learn that I'm not the only one, so Roy is also speaking about hematopoiesis. I don't know whether somewhere over there, I don't know. Okay. And of course you all know that hematopoietic stem cells make all these different cell types, red blood cells, megakaryocytes for making platelets, and then all cells of the adaptive and the innate immune, or with innate immune functions. And I was really interested to learn that actually most cells of the human body, if you just count them, you don't weigh them, but you just count them of hematopoietic origin, and that's due to the fact that actually more than half of the cells are erythrocytes already. And so in some, that's probably the stem cell system that produces the most diverse types of cells. And there's massive research, of course, that the hematopoietic stem cells sit in the bone marrow, was known more or less since the late 1930s. But I think, I hope I will manage to illustrate, if anything, that we still know many fundamental things about the system are not really well known, actually. So one of the interesting aspects here is that these cell types, of course, have very different lifetimes. That really make a big span here. People in this audience, Rob de Boer, others, Becker, have really contributed down here and here to really getting very, very accurate estimates of these lifespans. And you need to make them at the right numbers, and this is just a top of the envelope calculation for humans. And it's quite breathtaking. You might have a million erythrocytes per second, so every second you sit here making millions of erythrocytes that come from the bone marrow, and somehow the system has to do that. Why is it interesting to talk about it? If you open the textbook, it looks like everything's known. And this is a realization that really came with new tools in recent years. Is the main tool to study hematopoiesis has been transplantation into irradiated or conditioned hosts, either in human, of course, a mathematic application, but mainly in mice. So you take hematopoietic stem cells, label them, put them in the mouse. If you do this experiment, there's a very interesting outcome, and this has really become more clear through single-cell techniques, and it's an experiment that Hans Reimer did in his lab, and we discussed together, is that you just transplant a chunk of bone marrow that comes from a mouse, and you just take the cells out, okay, put them in an irradiated recipient, and you have very sparse labeling of cells, so there's just one labeled cell. So how will the labeling be represented in the reconstituted bone marrow after transplantation? And you already see this here extremely unevenly. Okay, so the situation where the labeling frequency roughly stays within sampling error in the actual reconstituted bone marrow is the minority of cases. In most of the cases, will actually get lost. In some of the cases, will be hugely overrepresented. We have cases where for this single cell makes up 50% of the entire bone marrow, which is telling us that during transplantation, even if you transplant many stem cells, not many of them will actually contribute to the reconstitution. So few stem cells produce output post-transplantation. And this is compatible with other types of experiments that, for example, use genetic barcoding of single hematopoietic stem cells that are then transplanted ex vivo. Okay, so we want to look at some of these questions that I'll pose here, but we will look at it for hematopoiesis that exists in the native normal, unperturbed state of the bone marrow without transplantation. So that's the goal for this talk. So we will not use transplantation models. They're very interesting, medically, but suddenly it's becoming clear that transplantation is a very special situation. So what does unperturbed endogenous hematopoiesis do in terms of how many stem cells actually produce output during endogenous hematopoiesis? How active are they? How active are the progenitor cells? And by active, I mean both in terms of proliferation and differentiation. We know that the stem cell number is maintained over the course of the life. It's not precisely maintained. There's some interesting slow dynamics during aging, but I think before we understand these slow dynamics, we really need to have a good model of understanding what in principle maintains the number of stem cells, and on top there's something slow drifts going on. And a large part of my talk, namely the sort of 1/3 at the end, will be devoted to the question, very pivotal question in the system. You have all these many cell types. What are the differentiation routes actually phenotypically that lead to these cell types? We will look at all these questions in steady state, okay, and not for perturbations, because of course many of us are theoreticians. We are aware of the situation. We need to understand the steady state because before we need to understand we can understand perturbations. And the idea will be, of course, this is very difficult to observe. So for many stem cell systems in the skin, in the hair follicle, in the gut, you can do imaging. There's few stem cells, they sit in a location, you can image progeny by using just a few fluorescent colors, and a lot of it is known about these systems. The bone marrow, of course, is a very fluid type of system, and imaging doesn't get you very far because the timescales over which you can image intravitally, of course, are not the timescales over which these processes will happen, as you will know, you will see. So we need to turn to other tools, and the idea is we want to get quantitative data, of course, that's self-understood, but we want to use models, and we want to use these models to infer properties and also go into further experiments. So that's, of course, the basic model for inference, and that's just simple population dynamics. This slide I just put up, so you get influx of cells here. They can proliferate, they can die, and they can differentiate. Just to remind you of the fact that this is although it's written down very easily and of course without much thinking, and in some sense it's probably an exact type of equation that all these processes are proportional to the cell number. It also creates a difficulty because that means how they go into this single rate constant here. That means just measuring cell numbers will not be enough to say anything about individual parameters. So we need to turn to other tools to find out things about rates, things about lineage topology. That's what the first part of the talk. That the starting point of this will be the second part of the talk. And of course, there's a lot of diversity if you look at single cell outcomes. And a very interesting question I will not be able to touch on is really what are the origins of this diversity. Okay, so the tool we will be using, and this is really through the chance contact with Hans Reimer, who's been a pioneer of this in the hematopoietic system, is fate mapping. And fate mapping is, as I already said, we need some additional information in addition to cell numbers. So we induce a label here, and then the label is genetically heritable. So it's different from labels like deuterated water or BRDU that people are often using in immunology. But it's a heritable label, so it will be passed on to the cells as they proliferate and differentiate. Okay? And of course, this, even if the system is in steady state, once I induce the label at a given time point, the label will not be in steady state, so it will be very useful information. And this is just my only real molecular slide. This is the work of Katrin Busch, who made, I think, 6 transgenic mice to fate map hematopoietic stem cells. 2 of them worked in the end, and that's the one that's working best. And I was pleased to know that Roy actually also made one, namely the FTG5 mouse. So just to mention that. So there's more around in the field now. This is coming up. So at the same time point, people have been producing these mice. And the idea is very simple. You take a mouse where you, and I think that's important also for the second part of the talk to understand the principle here, because in the second part of the talk we will put something more complicated in this locus down here. There's a reporter that is sort of preceded by a stop cassette here, and the stop cassette is flanked by LOXP sites in an orientation that this can be cut out by the activation of the Cree recombinase. And the Cree recombinase is expressed under the control of a promoter that is expressed in a number of cell types, but within the hematopoietic stem cell hematopoietic system, it's actually predominantly expressed in hematopoietic stem cells. And we were really lucky that this type of system, actually, as I will show in a couple of slides, only labels hematopoietic stem cells and none of the progenitors in the system. So it allows us to do fate mapping from the very top of the hierarchy. And then, of course, then the cells become yellow here. There's a YFP in this reporter. And all the progeny will become yellow. This is permanently excised. And the important point is the Cree is inducible. So you can do this at the time point at your will. Okay, and we will exploit that situation. So what do the data look like for this system? So let me just draw this sort of what people until about 5 years ago thought, and we still think, is the basic structure of hematopoietic differentiation architecture. You have hematopoietic stem cells. They have a pool of what are called multipotent progenitors that go under different kinds of names. Then there's some lineage bifurcations, but you still get what traditionally is called a lineage-restricted progenitor that produces mature cell types. And if you do that, you label hematopoietic stem cells indeed. So each dot is a mouse. So each mouse has a dot in every entry. These are the cell types here. At 1 to 3 weeks, you only see the label in the hematopoietic stem cells. You see a tiny fraction in some myeloid progenitors, but that's of no consequence. Here I can assure you. So the hematopoietic stem cells are labeled. You wait a little bit longer, and you already have to wait 1 month to see them coming up in myeloid-restricted progenitors here. So not only the stem cells are labeled, but also multipotent progenitors, myeloid-restricted progenitors. Some macrophages are labeled, or monocytes to be correctly. Then you see it in the lymphoid progenitors, but massively in mature myeloid cells. And if you wait very long, and really this is very long, it's a half a year or longer, you finally see some label turning up in the lymphocytes. So that's about the time scale of the system at steady state. There's one important fact here, and this was a lucky coincidence by constructing the system. The labeling frequency is quite small. I'm happy to comment on this with the aficionados, but if we believe that the stem cell pool is more or less homogeneous, which we know it isn't, but it's not a bad assumption for our purposes here, it means that we label about 1% of the stem cells. So we can do a limiting dilution experiment and lower stem cell labeling frequencies here, and ask the question, is there ever a low labeling frequency at which we do not see any progeny labeled anymore? So in other words, did we only label what people may call in the field quiescent stem cells? And the answer is no. We never see a mouse, no matter how small the labeling frequency, and sometimes 0.1% is about 10 to the 4 stem cells, so it's about 10 stem cells labeled. All of these mice produce differentiated progeny, which actually means that a lot of the stem cells in vivo are actually active. So let's now turn to the question of how active they are. We estimate it must be at least a third over 9 months. So several thousand stem cells are definitely active in vivo. Otherwise, we wouldn't get these types of results. And to do that, let's just focus on 2 adjacent compartments. So this is published work, so I'll go pretty briefly through it just to get the idea across. Say, for example, you look at the compartment of the stem cells themselves, and the next traditional compartment, which are called short-term hematopoietic stem cells, which really is a multipotent progenitor, and you see the labeling frequency here is high, and then it's rising, okay? You see also there's variability between the mice, but of course we have an internal control, so we can normalize the labeling frequency of the subsequent compartments with the labeling frequency of the stem cells, and turns out to be a useful thing to do. So now the question is, what parameters determine actually the rate of acquisition of the label by subsequent or by downstream compartments? And it turns out that the answer is very, very simple, surprisingly simple. If you look at the basic processes, so cells can die, cells can proliferate, cells differentiate from one compartment into the other, so that's the 3 things we look at. It turns out that the actual labeling frequency, so the ratio of labeled cells in a compartment, is governed by a very, very simple equation that is just saying over some time scale, the labeling frequency will equilibrate with the labeling frequency of the upstream compartment that's feeding it. So asymptotically, all the hematopoietic system, the labeling frequency will equilibrate to the labeling frequency of the stem cells. And how fast will that equilibration happen? Well, it will happen over time scale that's given down here. And this is, if you look at it, that's the rate of outflux of this compartment. So it only depends on parameters in this compartment, or can be written in this way. Outflux of this compartment. This is loss by cell death. So these are the loss terms. And this is what offsets the loss, namely proliferation within the compartment. And just mind you, if this were a stem cell, this would be exquisitely balanced. So that would be 0. This time would be infinite. Okay. So this somehow measures, and we call this a residence time in this compartment, this measures the degree of self-renewal in the compartment. So kinetics of label propagation measure how self-renewing a compartment is. So what does it look like in a concrete example? This is the compartment that just comes after the hematopoietic stem cells. These are now all the aggregated data from 100 mice and normalized to their internal controls. And you see they fall on a beautiful curve that's exponential. It's very, very simple, as would be predicted here, if the output of the stem cells were constant in time. And of course, now you could argue, maybe we did it wrong, maybe it's a linear fit is more appropriate and so on. So we just looked at data that come later, and that's shown here. And they more or less fit the curve, I think, within the confidence bounds of the experimental accuracy of the predictions that we can achieve. You can discuss a little bit about the very last time point. That's a Methuselah type of mouse here. And the question is a little bit whether hematopoietic output declines a little bit in old mice. But from this diagram, we would say for a large part of the lifetime, actually output stays at the rate that you would predict from young mice. Okay. So you can discuss already from this simple graph how good a model the mouse is for studying stem cell aging. There is stuff going on. We see stuff in the bone marrow that's going on with age, but it doesn't seem to affect very much the output. Okay, the other interesting feature is a number. And this number means that a lot of the delay that we see is already in the transition from the stem cells to the first multipotent progenitor compartment, which is itself almost self-renewing given the lifetime of the mouse. So the mouse lives about 2 years, a bit longer here sometimes. So at about a year, the mouse sort of would keep on doing hematopoiesis if the stem cells were actually switched off entirely. Okay? And this is something we were well aware of, and that's actually an extended figure 10 in the paper down here. We sort of made this prediction, and I think no one really cared. But there's now 2 experiments that actually showed it precisely. Okay, and that's been for us somehow increased our trust in this type of experiment, in this type of interpretation very much. There's people by Axel Roer's group, another experiment that showed now you can ablate hematopoietic stem cells in the mouse bone marrow without doing any other perturbation, and the mouse will be fine for at least a year. And it will actually even be fine if you do minor perturbations of the system, minor challenges. Yes? Right. Very well spotted. So the idea, and this is the whole simplification which we sort of got quite a long way, is that we look at the system in steady state. And of course, we really assume that these rates are regulated by feedback mechanisms of some sort.
Think about, I don't know, like a constant time that cells are pre-programmed to do the system.
How to think about that? Yeah, let's speak about the rates in the next slide, okay? But of course, the main assumption here, I mean, these rates will not be constants when the system is perturbed. That's very clear. So there will be constants if you look at steady state. And let's look at what the actual numbers are, because the numbers are quite interesting. They're down here. Some of them are really slow. So you can now do this label propagation here, and what you see is that if you go along the myeloid branch, the biggest delay is between the stem cells and the next compartment, and there's a smaller delay to what's called multipotent progenitors, and then afterwards everything's very fast in the myeloid branch, which is telling you that self-renewal is really decreasing along this chain. It's not so surprising, but we can now put numbers to it. But at the same time, we see that what the cells are doing, namely proliferating and differentiating, is continuously increasing. So at least in the myeloid branch, not so much in the lymphoid branch, you separate self-renewal from proliferative activity of the cells, and you may wonder whether this actually really prevents the accumulation of mutations in these self-renewing compartments. And this is what we estimate for the average activity of a stem cell. We would say it's active once in about 100 days, once every 3 to 4 months it will produce differentiated progeny. That's an average, assuming that all the stem cells in the bone marrow are active. And the interesting thing is that we do not see any real evidence of delay in our data. So this is really fitting an exponential curve. So there doesn't seem to be a really fixed waiting time. So with this simple distribution of exponentially distributed waiting times over this very long time scale, we do a reasonably good job, actually, interestingly enough. So, okay. So much for basic quantitation. You see other interesting things. You see how many flux goes in the lymphoid branch versus the myeloid branch is about 100-fold ratio over here. It's unclear whether this is selective proliferation or selective differentiation of these cells. So there's a lot of interesting questions that come up by this type of approach. I would pause for a minute here just to also stress very much that, of course, this inference here is only in any way meaningful if there's good statistics in terms of parameter estimates. And we tend to use a technique, it's called a profile likelihood. So it's a very useful approximation to the true likelihood. And with this, we can give confidence bounds of the parameters. And these are the profile likelihoods for the parameters as you've seen in the previous slide. And you see all of them are actually bounded. And some of them only have a lower bound, but the lower bound is meaningful in terms of its order of magnitude. So that means that these data that went into this here, which is mainly the fate mapping data and then some data about compartment sizes, are highly informative about these parameters here. So that also means we can use the model to make predictions, as I've showed here, with narrow prediction bands, and then people can do experiments. Okay. So these stem cells are rarely active, and they don't exhaust themselves. But nevertheless, if you think about 10,000 of them, it means about 100 of them are active every day in producing differentiated progeny. Okay, so about 1% per day. So the question is really also how stem cell numbers are maintained, and it's a very general question where there seem to be different answers for different systems, in Drosophila, in the gut, and so on, in the humans, and many other type of stem cell systems. And there's really two competing ideas about that. And one is that, and that's a very traditional textbook idea, is that cells undergo these asymmetric divisions, and it's really the hallmark what a stem cell is. It can sort of reproduce itself, and it can reproduce other cells. And the assumption here is that every division is actually doing this, and by this kind of self-autonomous mechanism, you sort of keep the stem cell pool, and it can produce differentiated progeny, and then you might be able to count the number of stem cells by the number of niches that you supply for these cells. In many other stem cell systems, in particular in mammals, so in Drosophila people have evidence for this kind of mechanism. And it's not, I'm not saying that it never happens. The question really is it a mechanism that determines compartment size? In mammals, very often what you find is a mixture of all of these divisions, actually with many symmetric divisions of stem cells and also symmetric divisions that lead to two differentiated progenies. And of course, this does not allow anymore for cell autonomous maintenance of compartment size. You need balancing mechanisms at the population level here. And this is extremely difficult to observe because you may want to observe stem cell divisions in the intravital imaging in the bone marrow. So in vitro, it turns out the in vitro experiments are all interesting, but the point is that they really activate the stem cells very much. So we don't have an in vitro culture system where we see the same slow rates of stem cell proliferation and differentiation. And it would be quite cumbersome to keep in the lab, actually, because I had to keep it for half a year or something like that. So we want to go in vivo. In vivo, this is very difficult to observe. We may observe a division that leads to two different types of progeny here, but the question is, do they really differentiate into different types of cells? And the time scale is quite long. So we reasoned we need to again go with inference, and we thought, okay, we need to know something about proliferation rates here that is distinct from differentiation rates. And so this was our fate mapping. We got a lot of info about differentiation rates, and it actually turns out we get something about what's called a net proliferation. But of course, classical assays like BRDU incorporation or dilution will give you information about the proliferation rate. So we thought, why not combine these two assays? And the reason why combining should work is that if you actually write down now the different types of stem cell divisions, they behave differently with respect to where the BRDU ends up. So symmetric self-renewing divisions, the BRDU ends up in the precursor in the stem cell compartment. Asymmetric divisions, it's divided, and symmetric differentiation divisions ends up in the progeny compartment, and so on. And the fate mapping label will behave differently. So we reason maybe we can use these two labels and try to make inference on the frequency of these processes. We wrote down the model in all of these processes, and I'm just showing you the result, that the inference actually works. We get estimates and confidence bounds, and this is, I'm showing you the most interesting inference here, and it's purely an inference from the numbers. And the inference from the numbers is that the stem cells divide symmetrically all the time. Okay? You can give confidence bounds to this, and they say, okay, they divide symmetrically at least 70% of the time. So the majority of divisions is symmetric. And this asymmetric sort of model is just a minority of stem cell divisions. That would be our prediction to the experimental community from this, which of course immediately implies that you need some kind of population level sensing for homeostasis in the system. And it also implies something that the medical field has been quite sort of interested in for a long time, namely if they find clone size imbalances in patients by some so coincidence, some maybe chromosomal translocation or so, people always get alert and say this is a pre-malignant sign of pre-malignancy. And okay, so I have 5 minutes for the next 50 slides. A sign of pre-malignancy. And but of course, in this model, clone size imbalances develop very naturally in the system without any sort of mutational advantage of the cells. Okay, so one needs to be very, very careful in interpreting these kind of data. Okay, so in the second part, and I think I can summarize this more or less in the 5 minutes I was given. I would like to start with the question, what is now the topology of the differentiation pathways downstream from the stem cells? And for this we need tools. And the interesting thing is that there's a lot of debate in the system, and a lot of debate comes through single cell sequencing, because people now observe states of single stem cells and they try to make inferences of what becomes, where these stem cells will develop. And we would of course maintain that you can't do that unless you make a lot of assumptions. So are these assumptions good or not? So let's look at fate mapping approaches to this. And the question really is, do we have this traditional tree-like structure, or is it more like a bush that the stem cell is already committed to producing only certain types of differentiated cell types in the adult bone marrow? And just sort of an advertisement of a lot of previous work that we did here, it turns out that the really informative data for this are single cell progenies. So you want to label individual stem cells and see where they go. And there's an interesting mathematical reason for this is that if you want to make inference, for example, you want to distinguish between a linear and a branch topology, the mean values can be entirely identical or indistinguishable experimentally. But as soon as you start to go to single cell progenies, which will show some variability, then you see that in the second moments of this distribution that you now will observe of single cell progeny outcomes, you see distinct differences. For example, you see a strong correlation between B and C here, and you see no correlation here. And it's difficult to formulate intuitive rules for that, so you really need mathematics to do that, and we wrote a little review about this. And there's a lot of work that is really being driven by Michael Flostoff and my group, who's now independent in Munich, about T cells, CD4 T cells, now CD8 T cells, CD4 T cell differentiation pathways. But I would also mention the work by others, Ken Duffy, Leila Perry here, and Roland Reger's group, who use similar ideas, not exactly identical ideas, but similar ideas exploiting single cell progenies using mathematical models and inferring lineage topologies. And let me just sort of outline the system that we're using here and showing one of the first results. So the idea is really one from Hans Reimer. And he looked at this confetti locus, and he can produce 4 colors, and sometimes he can produce a bit more colors and so on. But we have 10,000 stem cells. So we need much richer codes than we can produce by fluorescent labeling. And what he did is something very simple, and it's actually an idea that's been around in the literature. Also, Ken Duffy published on this theoretically, but Hans Reimer also was working on this for a long time already. So this idea emerged independently, and he's now been able to sort of really put it into practice, and it's working. The idea is you just take LOX-P sites and make a whole series of them in orientations that you can get either excisions of these orientations or inversions of opposite-oriented sites. And you do this, and you think under conditions of transient Cree activity, this system will operate for a little bit, and then it will sort of produce random outcomes. So this is our VDJ type system, except that we don't get junctional diversity. So here's my repertoire part of the talk, and I'll run quickly through it. It turns out it's a beautiful, simple repertoire. You can sort of calculate all the things that can happen, and you get about 2 million codes with these 10 LOX-P sites and 9 unique sequence elements between them that come from plant, so they're very identifiable. So about 2 million different codes. That's already great for 10,000 stem cells. But the important point is this is also a lottery. It's a bit like the immunoglobulin recombinations. Some are more frequent than others. And in this system, we can write down a Markov model with a very large transition matrix, 1.8 million by 1.8 million, and we can actually compute these generation probabilities. So we actually know what are good codes and what are not so good codes. And I will jump over this just by saying that about half of the codes that we find are what we think are rare codes. They're good, and we have a lot of experimental tests for that the model is actually doing fine. Okay. So if you now apply this framework, we will induce codes now. I'll show you just a maybe two brief experiments on this. In the end, in retrospect, I thought it was an ingenious experiment to not start in the adult. Well, maybe I'll come to that in 2 slides. But start in the fetus. Start at a moment in time at embryonic day 10 somewhere, I don't know what's written here, where the progenitors of the hematopoietic stem cells that will end up in the bone marrow appear in the fetal liver. Okay, so there's no other hematopoiesis present except these progenitors. And then we ask the question, if we label individual cells here, what will they do in the adult bone marrow? So it's a very long developmental process, but let's look at it. This is single-cell Sanger sequencing of individual cells. And we do find, not very often in this small, we do find rare barcodes here. So these definitely have been generated in single cells. And other may contain redundant barcodes, okay, other parts of the pie here. But what is interesting that we find them across the whole range of clone sizes. So some of these single cell HSCs have given rise to really massive clones in the adult bone marrows. Others didn't, or we didn't really sample them. And this would be clone size distribution that we infer from this data. It's very, very broad. It's broader than what's neutral drift. It's broader than exponential growth. And these are some of the barcodes that we found here in single stem cells. And this is looking now at the periphery, at progenitors, myeloid, erythroid cells, and lymphoid cells. And you see there's some that really produce all cell types. And we can go to more of these. For these, we don't know the codes in the stem cells because we just sequence about 1,000 of them, but we don't know in 2 different mice. We don't know the codes in the stem cells, but we know that they have been generated in a single progenitor. And you see most of the single progenitors give rise to all cell types in the mouse body. So at least from a fetal perspective, the stem cells are multipotent. But there's still and we see, I should also point out, we interestingly see some stem cell clones, and some of them are large ones here. This one here, somewhere, I don't find it right now in the time here. It's here. It's only producing erythroid myeloid cells, okay? So there seem to be clones that are coherent in their developmental potential, but most clones actually make all the cells, and you have some lymphoid clones down here. Okay. So how are these lineage pathways now related? Are there lineage pathways at all? And to this we just now turn it also again at the rare codes here. It turns out you don't need to restrict yourself to the rare codes. The signal is so strong that you can do this with all the codes without factoring generation probability. And you can ask, how are the codes used by 2 different peripheral cell types? And we reason if there's a large concordance in code usage here, then they must have to do something to do with each other. We don't know exactly what it is, but we find that erythrocytes and granulocytes extremely correlated in the usage of their codes, and also in the percentage of shared codes, while erythrocytes and B cells are much less correlated, and B cells and CD8 T cells are again strongly correlated. And you can now define distance measures. Both of them work, 1 minus Sperman or 1 minus overlap. You can group the cell types. And what you find is an extreme sort of division into 2 very distinct clusters. And it's extremely reproducible. And it's the classical clusters of the Weismann model is aries and granulocytes. And later we also have monocytes clustered together extremely strongly, and all the lymphocytes clustered together. And this is not a question of stem cells having already sort of restricted fates, because we can do this analysis for multilineage clones, so for stem cells that produce all the cell types, and we again find the same division. So it's hard to escape the idea that developmentally there is something common in the there's a common erythromyeloid branch and a common lymphoid branch. We're not really claiming this, but I think that would be a very simple explanation of the data. So in these branches, there should be common progenitors. Well, do we find evidence for common progenitors? This is a classical common progenitor called a CMP. And in this CMP, we find actually that all the codes that we find in CMP, we find in erythrocytes, and we also find in granulocytes, and they amongst themselves are very strongly correlated. It's hard to escape the idea that somehow this CMP is sort of a progenitor in the erythromyeloid branch, which is very much the classical model. And I'm a bit emphasizing this so strongly because the classical model at the moment is in the process of being destroyed from papers that come out from single-cell sequencing data. Okay, I think we have no evidence to abandon it from these fate-mapping data. Okay, I just want to say we can do this for adult hematopoiesis. It has its own problems, but we find exactly the same signatures also during adult hematopoiesis when we label adult stem cells. So this is the summary here. We think what we can know from these fate mapping data is that, first of all, the basis of self-renewing or nearly self-renewing cells is massively increased in the adult steady state situation. There's a lot more compartments and actual stem cells that are very, very long-lived. So there's a broad basis. These cells undergo acceleration of proliferation before they finally differentiate into lineages, and there seem to be linked fates in these type of things. And of course, there's many now important questions that we're sort of trying to address one by one using these tools about the regulation of the system, but also about the molecular basis. Thank you very much.
Questions? Questions?
Well, that was fascinating. So just focusing on the last bit, which you were going kind of fast on. So I think that you said that there's multi-lineage barcodes, in other words, that contribute to everything,
Right.
that nonetheless still have correlation between erythroid and myeloid, let's say. So do you believe that there are some stem cells that will, although they can contribute to everything, they have a greater tendency to go to one side or the other? Is that what you're saying?
That may be possible. We did sort of, as a null model, we just sort of simulated things, and we simulated a model where the stem cells have just go into this progenitor pool, and the progenitors have equal, so with some rates sort of, but each stem cell is identical, can go into one or the other branch. And this model is enough to generate these correlations. So it may be that I'm not saying this. What you said may be an explanation, so that they actually have some bias. But it may also be that they have zero bias, which I don't believe. But the correlation structure is simply imparted by how long lineages stay together in a developmental pathway. And we don't know the answer.
Right, because the stem cell is only ever active once in 110 days.
Right.
Is that basically the
On
story?
average, yeah.
And it's generally coherent. It generally does symmetric division.
Yeah.
Is that the derivation of the? Could that be the reason why you can actually seem to see that? You see what I'm saying? Yeah.
Yeah. Maybe we should discuss this in the break, because we have a lot of ideas on this, and it will steal time. So, but yeah.
I wonder if you have some thoughts on where the single-cell sequencing papers are going wrong and interpreting their data beyond the result.
Right. So first of all, I would say very categorically, despite all the attempts to put lineages through single-cell data, this is, of course, only hypothetical. There's no evidence in the data that this is a developmental lineage. And but of course, in some cases, it will be very informative. So I think that's one of the directions we want to go now, talking with these people. But I think the really unresolved question in this single-cell field is how much of the variability you see is a breathing of the regulatory network, and how much is a unidirectional development. And just to give you an example, we have an IL-7 receptor fate mapping mouse that labels lymphoid cells. And you actually label a certain fraction of the stem cells with that. That means they, at some point, had all IL-7 receptor and the machinery with it probably, so they were appearing lymphoid biased. These stem cells produce happily myeloid output. So, okay.
Last question.
Thanks. Oh, lots of data. You made very short notion that clonality of hematopoiesis is might be a derivative of chance, while clinically there are plenty of papers, and they are coming faster and faster, that clonal hematopoiesis, and especially the myelodysplastic syndrome, precede leukemia. Have you taken your system already into the older animals or the pre-malignant, or you might note about it?
So I think that's on the plan. We want to do clone analysis. We haven't done that. We can do this with the barcoding system now. And the only thing I'm saying is one needs to be very careful in interpreting these data. I'm not doubting that you can get expanding clones by proliferative advantage or some other type of advantage, competitive advantage. But the question is, you will also get clones, large clones anyway. So the key is really distinguishing between the two, I think.
This is yours, or this was here?
No, that was from the NCA class.
The laptop's over there. Okay, thank you. Do you want to use this to have a... Right, so the next talk is by Kim Jacobson, which will talk about the role of histone-modifying complexes in regulating B-cell programs to infection.
Do you have the pointer? Is the pointer? Oh. Is there a point? Do you have the pointer?
Pointer should be... Alice, can we take the pointer?
Thank you. All right. So this talk is mainly a cellular biology talk. So basically, our lab is interested in how B-cell fate decisions are regulated. So we look at B-cell differentiation during immune responses and really focus on the molecular regulators of how germinal centers, plasma cells, and memory B cells are formed, and then how memory B cells respond during secondary infections. We focus primarily on transcription factor networks, as well as epigenetic regulation, which will be the first part of the talk. We're also interested in how these processes go wrong, and particularly during chronic infection. There's been this, in HIV and malaria patients, there's been a population of cells called atypical memory B cells, and some people have raised this in questions during the week, where we know that they're elevated in these patients, but there's still kind of a lot of hypotheses going around about their function or dysfunction, and so we wanted to look at this in vivo. So just to introduce epigenetic regulation, so as you know, a lot of programs during cellular differentiation are driven by transcription factors. There's also another element of regulation which doesn't affect the DNA sequence, in that by modulating chromatin conformation, you can either access or restrict transcription factor binding to DNA, and that's another element of regulation. And this is the question for Uri, who's not watching. This is the only stochastic part of the talk. Is this conformation stochastic or predetermined? Yeah. All right. So we primarily look, so there's a number of different types of epigenetic regulation, and we primarily look at histone modifications. So on the tails of particular histones, there are modifications that can be made, and there's kind of three different types of modifiers: writers, erasers, and then the readers that bring in the transcriptional machinery to read the code. And basically what this does simply is either put chromatin conformation in a euchromatin, or which is open and can allow transcription factors to bind, or a heterochromatin, which basically closes it up. And the reason I got into this is to look at the memory B cell problem in B cell differentiation. And why I say problem is because in B cell biology, we know a lot about the transcriptional programs that regulate differentiation into germinal centers, and transcriptional factor programs that regulate plasma cell differentiation, but not much is known about memory B cells. And I started on this a long time ago, and I've gone through a number of labs trying to work out what factors are involved in memory B cell differentiation. And there's certainly, what we found is actually memory B cell gene expression is quite similar to naive B cells. So there's not this kind of unique transcriptional program such as there is in germinal center and plasma cell populations. So we decided to look at whether there's, because memory B cells are intrinsically different to naive in terms of their ability to respond much more rapidly during secondary responses. So we decided to look at whether there is epigenetic regulation rather than gene expression transcription factors. So we basically just picked a histone modifier to look at. Our collaborators were working on a histone modifier, a histone acetyltransferase called MOS, which is important for long-term repopulation of hematopoietic stem cells. So we used a gene-specific deletion in B cells to look at the role of MOS. And just to summarise this, because this was published quite a few years ago, what we found is that when we deleted MOS, we had a 5-fold decrease in the dark zone, but not the light zone of the germinal centre. And this correlated, I should say, to output of the memory populations. So plasma cells were actually unaffected by deletion of MOS. The numbers of switched memory B cells were okay, but the affinity was reduced. And we had an increase in IgM memory B cells. And then this correlated with differences in secondary responses. So we had an increase in secondary germinal centres, but a decrease in high affinity plasma cells that were formed. So this was kind of just a proof of principle. If you delete an epigenetic modifier, you can affect the outcome of the memory population and secondary responses. But basically, we wanted to go back and do a much more detailed, or get a much more detailed understanding of how epigenetic regulation is important during B-cell differentiation and fate outcome. So to do this, we obviously had to pick particular modifiers to look at. And in the stem cell field, much more is known about the roles of different histone modifiers. So there are two main groups that people tend to look at. One is the polycomb repressor complexes, and they're important for repression. And the other is the trithorax group of proteins, which is important for an activating mark. So we decided to take a look at particular molecules from those two groups. A lot of work, it's not here, but a lot of work has been done on EZH2 by 3 or 4 groups, and the critical role in B-cell development as well as B-cell responses. So another reason we decided to look at these two groups is the putative importance of the marks that they make on immune cell function. So a lot more work has been done in the T-cell field in this area. Basically, the modulation of H3K27 trimethylation, which is the repressive mark, and H3K4 trimethylation, which is the activating mark, can correlate with B-cell behaviour and chromatin accessibility during immune responses. So in CD8 T cells, for example, there's a lot of genes that are important for proliferation are often bivalently marked, so they have both marks in naive T cells, but the repressive mark comes off quite quickly after activation. Whereas immune function genes that are associated with either memory or terminal effector CD8 T cells often only have the repressive mark, and then during activation, during stage-specific activation, the K4 activating mark comes on. So modulation of these two marks has been correlated with important immune function. And this is just an example in B cells where you have the gene encoding AID. So in naive B cells, you don't have the activating mark, but you do have the repressive mark. Whereas in germinal centres, it's the opposite. You have the K4 mark, but no K27. And as you'd expect, you have then open chromatin accessibility correlating with the K4 mark in germinal centre B cells. So to look at these, we picked a few of the genes from the complexes. So MLL1 is one of the main molecules that makes the K4 trimethylation mark. And this, I'll bring this back up later in the second part of the talk. It forms a number of different complexes. One of its main binding partners is menin, and those two get dysregulated in a lot of different types of cancers. But what other partners is context specific. PRC2, polycomb repressor complex 2. As I said before, a lot has been done on EZH2, but EZH2 can actually be, the deletion of EZH2 can be compensated by EZH1. So it's not quite clear whether those studies that are looking at EZH2 are really getting rid of the whole complex. They do get a reduction in K27, but it's not completely absent. Another thing about PRC2 is that it brings in polycomb repressor complex 1 to enact gene transcription programs. And again, there's actually two types of PRC1, a canonical and a non-canonical. So we're looking at the canonical BMI1. All right, so we started looking at EED mice first, so PRC2, just purely for the fact that we had those mice first. And we're deleting specifically just immature B cells. And just briefly, we've done a number of different types of immune responses to really try and figure out if there's context-specific function or whether there's generalized function that we can make conclusions about. So similarly to EZH2, when you delete EED in B cells, you lose all ability to form germinal-centered responses. So that's quite clear at day 7, and you can see that there's absolutely no GC responses in these mice. This is in an MPKLH immunization model, I should say. Again, as you would expect from the lack of germinal-centered B cell responses, any IgG1 memory B cells is completely abrogated. And I'll also point out in the heterozygote mice, they seem to be fine. So one allele is fine to be able to form the complex, but if you lack both alleles, it completely abrogates the response, and same for plasma cell response. So long-lived plasma cells are completely absent. And it's not just germinal centers. So up until recently, with the EZH2 field, a lot of focus was on germinal center specific responses, but it's not just germinal centers, it's B cell, somewhere early during B cell activation that this complex is required. So a T-independent immunization with MP5-col, again, the response is abrogated. Now, some of these modifiers are important in a context-specific manner, so they might be important in one type of immune response, and this is Th2-like response. So we wanted to investigate whether it was also critical in a Th1 cell bias response, such as influenza, and it is. And you can see here, basically, there's no plasmablasts, there's no germinal centers either. So EED is required to mount B cell responses, but they can be activated. So if you put them in vitro, B cells from these mice, they are able to be activated. Now, another question, as I kind of alluded to at the start, that we look at in the lab is whether there's differential requirements during chronic infection. And just after we did the experiments I'm just about to show you, these papers came out in Science at the end of last year, I think, where they were looking at the epigenetic landscape of T-cell exhaustion. So a lot of work has been done, particularly in the CD8 T-cell field, looking at basically how T-cells become dysfunctional during chronic infection. A lot of that has to do with PD-1, but also other molecules are involved too. And what they found is actually kind of looking at it from the problem of, a lot of these checkpoint inhibitors, blockade have been used in cancer patients at the moment. It's the hot field. But a lot of these patients rebound, so they respond quite well to the blockade, but then cancer comes back, basically. So they wanted to look at whether there was differential regulation, something different about the chromatin structure in these cells. And they did. They found that there were kind of changes that were locked in. So we use a model in the lab to look at chronic infection, which is an LCMV infection. So there's acute and two different versions of this virus that can either give you an acute resolving response or a chronic response. And so when we infected these mice with the acute version, again, you got much reduced germinal center responses. But when we infected these mice with the chronic version, we actually got quite good germinal center responses. So, but little isotype switching, actually. So there's something about these GCB cells that are able to overcome this requirement of EED and potentially the K27 trimethylation mark, although we've got to look into that. And just to make sure that it wasn't something particular about that model, we also looked at a response to malaria. And again, when you can see actually up here, the mice actually have trouble clearing the parasites up until about day 20, which is where germinal center responses come up in these mice. And here again, so early in the response, you do have a reduction in GCs, but they're able to catch up by the time that germinal centers are established properly. So it's not just the LCMV model. Now, we have a couple of hypotheses that we're looking into as to why this is. But at the same time, we wanted to kind of ask the question, is this specific just to this complex? What happens if you look at the reverse? So what happens if you delete K4 trimethylation? And I guess, unsurprisingly, germinal center responses when you delete K4 are also reduced, because these two marks, even though one's activating, one's repressive, they're both important for modulating different B-cell programs or gene expression programs. But interestingly, we didn't see an effect on plasma cells, and we actually got an increase in the plasma cell population. And then surprisingly, when we infected the MLL1 deficient mice, we got the reverse effect to the EED mice. So even though in the MPKLH response we did get a reduction in germinal center responses, in the acute version of LCMV, the GCs were fine in the MLL1, but in the chronic they were reduced. So, one, I guess, big open question, kind of like a big flag to us, is whether this is due to type 1 interferon signaling, which is known to be different between acute and chronic responses. So that's basically where we're going, is to look at the signals that are involved in modulating these complexes. And also, what is the landscape? So, kind of looking at this with a blunt object, I guess, in that, we're just deleting it completely in mature B cells. We don't know what other histone marks are there that might be important for these responses. So, in talking about signaling, we also are really interested in the idea of location-induced epigenetic regulation, and particularly within the germinal center. So in other systems, we're kind of used to looking at B-cell differentiation linearly, but in other systems, the epigenetic regulation is quite dynamic. So if you move into a different location, those changes and those complexes can change and modulate and regulate fate based on where they are. And that's obviously to do with the signals. So we're quite interested in looking at this within the germinal center. And there was a paper 15 or so years ago that was just doing histology on human germinal centers that showed that the polycomb repressor complex 2, like a lot of their components, are localized in the dark zone, but a lot of the components of PRC1 are localized in the light zone. Now, there's been, Ari Melnick has done a lot of work looking at the role of this complex and the non-canonical PRC1 in turning on the germinal center. But interestingly, BMI1, which is in the canonical version, is repressed mostly in the germinal centre, but in these papers seem to come on in the light zone. Also, if you look at RNA sequencing of plasma cells, BMI1 is actually highly expressed in plasma cells, and particularly, so plasma cell populations can be separated into BLIMP1 intermediate and BLIMP1 high, and BLIMP1 high cells tend to associate with long-lived populations, and BMI1 was most highly expressed in the BLIMP1 high cells. So, you know, similar to the model that you just heard in the previous talk, we're using a tamoxifen-inducible BMI1 reporter to try and figure out what populations BMI1 is expressed in and when. And we're still kind of working on it, but we've found that BMI1 is not expressed in memory B cells, but it is expressed in a subset of plasma cells. And it's mostly not expressed in germinal centres, but there is a small population that do express it. So this is actually, we've just started working on BMI1, but I thought I'd put this slide in. So unlike EED or MLL1, when we delete BMI1, we have no change in germinal centre formation, and that would fit with kind of all the expression patterns that we know. But interestingly, and potentially, hopefully associated with the expression that we've seen, when you delete BMI1, you get a large reduction in plasmablasts. And this is at day 7. So it's a mix of the plasmablast population at that time is a mix of short-lived plasmablasts from the extrafollicular response as well as the ones emerging from the germinal centre. So obviously we're going to do longer time points to really dissect that out. So just to summarise this part of the talk, you know, we're looking at a number of different complexes and how the dynamics of those complex formation in both time, so temporal and spatial locations, affect B cell fate decisions. And, you know, we've kind of just using a CD23 CRE, which deletes in all mature B cells, but we're now trying to look at stage-specific deletion with a little bit more sophisticated models. But I guess the open questions for the research itself would be basically what is the chromatin landscape in all of these different populations. We know a fair bit about naive and germinal centre B cells, but not much is known about memory. We've done a little bit of work on long-lived plasma cells in the bone marrow as well. But also how is that modulated in the absence of these different molecules? And importantly, what are the signals that induce these changes? That's really important, and that's something that hasn't been looked into much detail in our system anyway. Kind of on a broader level, how can we deduce how and when plasma cell fate gets locked in within the germinal centre? So we do know obviously a lot about what happens within the germinal centre. We know the transcription factors that are important to be turned on into plasma cells, but when and where the B cell within the germinal centre makes that decision to differentiate and make the choice between memory B cells and plasma cells is still kind of an open question. And what we hope is that by being able to kind of determine plasma cell fate within the germinal centre, we're able to go back and deduce what's happening or what's not happening in the memory B cells. And then just broadly, whether there's parallel mechanisms regulating chronic GCs in different disorders. And so I'll just go into the second part of the talk, which will be shorter. As I mentioned, we also look at transcription factors. And one of the transcription factors I've been looking at for a while now is one called c-MYB, which is important in B cell development. We showed a couple of years ago that it was critical for the long-lived plasma cell population in the bone marrow, for the migration to the bone marrow. But as part of that, working on c-MYB, we did gene expression analysis, trying to figure out why it's critical for migration. And kind of an interesting thing came up in that Tbet and Tbet-regulated genes, which are not usually expressed in this type of response, so a Th2-like response, came up in these cells. So here you can see, so K27, which is the repressive mark, is usually on in germinal center B cells from this response, repressing this type of response. Why this kind of grabbed our interest is because I think Andrew mentioned, isotype switching is regulated by different cytokines, which is obviously influenced by the pathogen-induced microenvironment. And we've known for a long time that different isotypes get segregated depending on different types of pathogens. But no one really understands, no, I shouldn't say that. So a little bit of work has been done in trying to dissect out the transcription factors that are involved between the cytokine-induced signals and transcription, but no one really knows how kind of that flexibility is set up within B cells to respond to different types of pathogens. It's also kind of quite important is because TBET expressing B cells now have been identified in a number of different disorders. It's unclear whether kind of the same cell is the same one in chronic infection, aging and immunity, as well as during normal viral infections. That's being dissected out. But what's clear is that knowing how to modulate TBET B cells will be quite important. So just to show you some of the data, IgG2C, which is downstream of TBET, was turned on in these mice. So normally during MPK-LH reaction, you do get some. It's not completely absent. You do get a number of different isotypes. It's just that IgG1 dominates. In the absence of CMIB, you can see you get a lot more. And as expected, this response was rescued by deletion of TBET. In these mice as well, we had another phenotype that we were looking at, which is within germinal centres, we were getting an upregulation of plasma cell transcription factors. So normally, if you look at a germinal centre population, you only get a small frequency, very small frequency of cells that are expressing a plasma cell marker. But in the absence of CMIB, this population increased, and you can see here BLIMP, which is turned on in plasma cells. We could find a lot more within the germinal centre. And interestingly, the upregulation of TBET was the reason why we were getting this. So when you deleted TBET, you also rescued this phenotype. So, all of that was in an MPK-LH response. We hypothesized that during an influenza infection, which normally turns on TBET and IgG2C, you wouldn't see any difference, but we did. So in the absence of CMIB, again, you got these what we call GCPC type cells. And again, deleting TBET rescued this phenotype. So it seems like it wasn't just a kind of CMIB turning on or off TBET, that it's a graded effect. And then just briefly, we were looking at the mechanism of this, and we found that upregulation of CXCR3 by TBET was the reason we were having these plasma cells kind of stuck in the germinal centre. CXCR3 is important for migration to inflammatory sites. So I'll just skip that. What we haven't published, though, is that it wasn't just TBET that was modulated. So we had a decrease in NFR3, and NFR3 has been reported to be important for IgE. And we found, similarly to NFR3 deleted mice, CMIB deleted mice also have an absence of IgE, at least in vitro. So, the data has kind of led us to this question, which is, how do B cells integrate signals to respond effectively to different pathogens? Because these cytokines are not induced in isolation. The B cell has to integrate signals and respond most effectively to each environment, and it's not all or none. It's not just one isotype is on and the rest are off. So there's modulations, and we're looking into now what the molecular mechanism of this is, how CMIB modulates between different programs. and potentially different isotypes. And linking it back to the first part of the talk, in CD4 T cells, CMIB has been known to complex with MLL1. And so we're potentially looking into whether MLL1 has this context-specific role. And I'll just finish up by thanking the people who did this work. So Dana was involved in the second part of the second story. And the first part has been done by mainly by two really talented honours students, Rohir and Cal, and is being continued by my postdoc, Andrea. And thank you.
Okay, questions?
Very nice work, Kim. Can I just start with the EED story?
Mm-hmm.
And you mentioned that those B cells respond, you said, quite normally in vitro.
Yeah.
And I just wondered how far you'd push them in terms of switching or differentiation. It
Yeah.
seemed a bit surprising given the in vivo chronic phenotype.
Yeah, so we have, we looked at differentiation switch. I mean, we just did basic 3 or 4 day stimulation, and they seem fine. And I was using that as reasoning earlier on to show that, well, it must be a germinal center phenotype in vivo, that if you stimulate them in vitro, they're able to differentiate to plasma cells fine. But now, because of the T-independent responses, I don't know if that's true. So I guess what I'm trying to dissect out with the chronic infection as well is how much, like whether the strength of the signal is
Right.
important and the type of signal.
Right.
And I potentially think that's true.
Right. And have you looked at the effect of MIB on cytokine production?
Cytokine production of B cells themselves?
For example.
No.
Okay.
But, especially if, so we are missing the link between if it's a direct effect or if it's an indirect effect. So, I mean, the direct effect is obvious that it's either binding to T-beta or NFR3, but potentially could be indirect and be modulating cytokine production, but we haven't looked at that yet.
Anyone else?
In the first part where you show the difference between the acute LCMV and chronic LCMV, the time points were you showing, were they, like, was it the chronic time point for the chronic and the acute time point for the acute? And so if you kind of tracked the chronic infection at different time points, when does it recover, I guess, is the question.
Yeah, so we've only looked at 2 time points, but it's the same time point. So, I mean, the chronic infection is, I don't know, it's still only, I think T-cell people only go out to 4 weeks, which for us is not that far. But we've only looked at day 10 and day 14. We haven't gone out to day 28 yet. So
you already see the difference.
Yeah. So
even at that's
so early on, so in acute and chronic infection, so there's different stories on this. So in the T-cell field, they've shown quite early differences, but in type 1 interferon signaling. So that happens actually very early. And I think by day 14, the PDCs are gone from chronic, in the chronic LCMV model already. Not sure about acute. There's a whole bunch of different stories on the B-cell side, which I won't go into, but they're suggesting the signaling is the opposite of what the T-cell people do. So it's still kind of a bit vague, but we definitely want to go into later time points. And also we're tracking antigen-specific responses as well, which are a bit different in those responses as well.
Nice.
So for the last talk of this session, Eilon Sherman will talk about a single molecule view of immune cell activation.
Okay. This is going to be a slightly different talk, using different methods than the ones that you've seen so far. And these are the people behind the data. So this is my group, and I'll be talking about data acquired by these two Yairs here. And I would like to thank my lab members, collaborators. Some of the data goes back to my postdoc term with Larry Simonson at the NIH, NCI, and some funding. So this doesn't move. So to mount an immune response, T cells probe the surface of antigen-presenting cells, right? And they look for cognate antigens. And this is how it looks like. So here are the cells. And what you see are calcium flux here within the cell. This is the APC. This is the T cell. And what you see is that the T cells actually respond within seconds, right? So the response is extremely fast, and it's highly specific and also highly sensitive. Do you know how many peptides are needed for cognate response? So number go down to 1, 2, few dozens. It's really under debate. And the point is that if this is the interface between the cells, then you have here the signaling complexes. This is the antigen presented on the T cell receptor, recognized by the T cell receptor and presented on the MHC. And here you have nucleation of different proteins. And these are the computational units of the cell. So the signal propagates here from phosphorylated motifs on the TCR through a kinase called ZAP70, phosphorylate SLAT, linker for activation of T cells, and these get phosphorylated and leads to multiple cell functions. So I think that the question still persists how the TCR peptide MHC affinity translates into these properties of immune response. And I think that we still don't really know. So our approach is to take the cells and label fluorescent proteins within. And then we drop them onto cover slips. And the cover slips are coated with antibodies that can stimulate the T cells. So they spread and they form what is called the immune synapse. And this is how it looks like. Hopefully you can see there are dozens of different proteins that come together, all nucleate in different clusters, and many of them are highly phosphorylated. So hopefully you can see the images and the colors. Obviously we can let the cells spread on cover slips that do not stimulate the T cell receptor, as you can see here. But still, you have very nice spreading, and you can see the different molecules without the phosphorylation. So the question is, basically, you have these different complexes, right? And these are complexes that are nucleated by LAT, linker for activation of T cells, and you have different enzymes here, PLC gamma, GREB2, Cbl, SLIP76, and all of these would give rise to different cell functions. So what we want to do, realizing that these complexes can be highly heterogeneous and very dynamic, we would like to resolve these in single molecule detail. And how do we do that? So this will be the first part of my talk, and then the second will be how we use super resolution imaging in order to understand better mechanisms of T cell activation. So some 150 years ago, Abbe told us that 2-point emitters cannot be separated if they're too close. This is called the diffraction limit of light. And basically, it's about 200 nanometers for visible light. So you cannot separate two proteins. And especially at the cell membrane, these proteins, they're what is called point thread functions, the blurring on the detector, all of these overlap, so you cannot separate these different emitters or different proteins. However, imagine that you could have just one point emitter, one protein at any given place at any given time. So this would give rise to this blur on the detector. This is called an iridescence, doesn't matter. It's about 200 nanometers in size. And realizing that there's just one emitter, say this is a GFP protein of 2 nanometers in size, this function would be symmetric. So you can find the center line of this distribution, and this would be the perfect location of your single molecule. Now going back to the cell, you have these different emitters that overlap, and you want to separate them. Now suppose that you could. So you light up only a few, and you tweak down the concentration of these molecules so you can really separate them. You localize them, bleach them, and then move on to another set. Stochastically, they come up, and then being assigned, you end up with a map of single emitters, single molecules, with resolution much better than the diffraction limit of light. Okay, how do you do this trick? So this goes back to these fluorescent proteins that can be photoactivated. One of them has been demonstrated in the lab of Jennifer Lippincott-Schwarz, and they realized based on these fluorescent proteins that can undergo switching between a dark state and a fluorescent state. Still, you need to excite here to get photons. This is called photoactivation. Using this trick, you can induce blinking of molecules, and then you can separate them in space and time and localize them. And by now we have different proteins, fluorescent proteins, that one can mix and match, photo switching between different colors, etc. So you take these proteins and you label your molecule of interest. And this is how it looks like. So this is a fixed cell, and the molecules that you see are called LAT. And this is just the plasma membrane. This is the footprint of the cell as it adheres on the cover slip. And what you see are the single emitters here. They blink. So we can localize them at any given frame. And then we take all of these locations and we collapse them onto a single image. And now you have seen this already. You see the clusters of the different molecules. Only now you can zoom onto any of these clusters and you can start counting molecules. Okay. The resolution that we get is about 20 nanometers, about 10 times better than the diffraction limit of light. And this is the localization error that peaks at around 10 or 20 nanometers. Okay, so what do you do with this kind of data? You want to infer some of the mechanisms that lead to T-cell activation. So one way is just to understand how they cluster in space. And this is called the pair correlation function. Basically, you measure the distance between every two molecules that you detect. And this is a control for randomness. Okay, so we run Monte Carlo simulations, and you see that the curve here exceeds this 95% confidence interval due to randomness, meaning you have significant clustering. And the clustering cannot be detected by diffraction limited microscopy, since much of it is below 200 nanometers. Okay. Also, you can employ clustering algorithms, as you've seen before in other talks, but here we can now use this to count the molecules in each cluster, and this is a cumulative probability distribution, and we realize that most of the clusters are really tiny, as small as single emitters or dimers or trimers, etc., accounting for about 80% of the data, and then only a few that catch your eye here are really big and have dozens of molecules. Okay. Now we can either activate the cells or not, and we see basal clustering without cell activation here, as you can see here by the green plots. But surely upon activation of the cell, you have higher level of clustering, and the clusters tend to be larger in size. Okay.
So the cells have CD8?
So these are CD4 T cells. Yes. And now we can actually monitor the extent of interaction with different effector proteins. So LAT is known to bind PLC upon its phosphorylation. And so this is an effector protein in red. And you see nicely mixing within clusters. And we capture this by statistics. This is called the bivariate pair correlation function. So you measure the distance between every green and every red protein. And the model here is of random labeling, meaning that you erase the color, and now you relabel randomly all of the points that you detected here. If the color did not affect the position, then you would see these curves, okay? And you see that red and green nicely mix within these clusters. However, for unrelated molecules like LAT and TAC, TAC is the alpha chain of IL-2 receptor, you have no interaction at all, so the correlation is flat. And there's some, so ZAP70 is the kinase that comes from TCR zeta and phosphorylates LAT, you have partial mixing, which is interesting. Okay, so not only that, now we can visualize 3 different colors, and with this you can start asking questions about cooperativity in these complexes. So you can ask, well, we know how to understand the interaction between red and green by the bivariate correlation function, only now we can ask how the presence or the proximity of blue proteins affect this interaction. And what you see here is a measure of cooperativity. So you see that all of these molecules kind of co-cluster and participate in these complexes. However, there's a lot of cooperativity within. So with this, we kind of come up with a model of having basal clustering of receptors and LAT proteins at the membrane. These diffuse and interact and coalesce into larger clusters upon TCR activation, and then nucleate signaling complexes, and these are really the computational units of the cell. These complexes would relay the signal into different pathways. They would amplify the signal as well. And you can now compare these to the cartoons that one gets from biochemistry. So this is what we know about the complexes, but now we see that there's a lot of patterning within. So now I would like to switch gears and to talk about how we can use these techniques to resolve mechanisms of TCR activation in single molecule detail. So,
excuse me, and
yeah.
how many TCRs are there in these large clusters on average?
Okay, so this is similar to LAT, and people say, okay, we have clusters with a mean, with an average number of, say, 60. And my claim is that this means nothing. Why? Because the distributions of copy numbers, of sizes of the clusters are really skewed, okay? Meaning that they are dominated by very tiny clusters, as small as monomers and dimers, and then you have only a few that have very large clusters. And they can have different functions, right? So who knows which is dominant? Okay, so bear with me. Now we would like to talk a little bit about these. So I'd like to talk about these mechanisms, and people don't really know once there's a recognition of cognate antigens, what is the signaling cascade that goes on. And basically there's a model, or these are models that claim that, maybe you have this segregation of these bulky proteins called CD45. So these act as phosphatases, they're glycoproteins. They're bulky, so they need to be excluded from this tight interface between the receptor and the ligand, maybe. And another mechanism would say that maybe there's clustering, obviously you saw that, but maybe there's some cooperativity in interaction. So maybe not all of the TCRs need to see cognate antigens, but only a few, and then they would send the information, propagate the information within the clusters. So let's start with this. Can we see that? So hopefully you can see, again, the movie. And what you see is CD4 in red, CD45 in red, and TCR in green. And this is a cell that drops on a cover slip, and we label only or visualize only the plasma membrane. And you see that these receptors segregate from CD45. Let's look again on just a single contact, and this would be maybe 1 microns in size. And you see that they segregate. And also you see this dark zone between them. I'll get back to this in a minute. So, to understand this better, we simulate the interface using agent-based modeling and Monte Carlo simulations. And we embed proteins within and let them diffuse and interact. So you have some interactions between them, some attraction between receptors and ligands. You have spring-like repulsion by these bulky proteins, CD45s. And then you have membrane elasticity. Never mind. So basically, what we now can do is we can take the single molecule images and compare them to our simulation. And if we understand properly the mechanics or the physics of the interface, maybe we can predict the location of the proteins. So basically, we take the green proteins from the experiment and we try to predict the positions of red proteins. And you see that we can nicely recapture the interface or the patterning here, the segregation between red and green proteins, also here. But not only that, we can actually follow the dynamics of this interface, and you see this segregation between green and red. This is the bivariate correlation that drops below 1, so this would be the segregation. And we follow this segregation over time, and we see that it saturates at around 200 nanometers here over time. So it takes a few seconds. And now using this model, we can actually look at different conditions. This is membrane elasticity or flexibility, and we can match exactly the dynamics of the interface, the segregation, and we can actually use this to probe some parameters that we cannot approach or we cannot know. And basically, these are the most favorable conditions. So we see ligand density, membrane rigidity, and the whole footprint of these interfaces or the contacts. So going back to these models, so yes, we see this segregation. Now what about cooperativity? Can we see this? So this is another movie, and you see the receptor here in green again. So TCR is in green. And now red is a kinase called ZAB70. Again, it binds phosphorylated TCRs. And what you see here is that at specific places, in specific times, you see recruitment of ZAB70 indicating phosphorylated TCR zeta. And these are activated TCRs. So the point is that this whole field is uniformly activating the TCRs. So any green point here has the same probability of attracting red proteins. However, this is very different than what we see. We see localized activation of TCRs, and it's highly synchronous. So we think that this is a hallmark of cooperativity. And these interactions, these clusters within clusters are actually maybe 200 nanometers in size. So these are really far distances. These are not direct interactions between proteins. And for that, we use, again, the models to try and capture this. This is a correlation function, again, indicating clustering within clusters. This is the model here. And we use the models in different ways. Most of the mechanisms that we tried to model failed. However, only 3 survived. And you can see these clustering within clusters, this localized activation. And they share long-range cooperativity by some mechanisms. We can talk about it if you ask me. Or this localized activity of LCK. LCK acts like a gate in a transistor. Maybe if it's bound, if it's trapped in a cluster, it can enable phosphorylation of receptors within its vicinity. So this could be another mechanism of the patterns that we see. But now we can summarize and say that we see, yes, we see microcluster formation, we see phase separation between these receptors and these bulky proteins and cooperativity. And with this, I'd like to conclude. And here I show you that we can move from these cartoons given by biochemistry to really single molecule imaging of individual clusters, individual molecules within clusters, and these are highly patterned. I showed you that we can use hybrids of experiments and simulations to understand the mechanics of the cell interface. This is just one contact of the cell, microvilli that touch the interface. And then you have this segregation of proteins, the bulky proteins from the tight interface. And then I showed you that there's some indication of cooperativity within these clusters. So not all of the receptors need to see cognate antigens in order to respond. So thank you. And just a comment, we have open positions if anyone is interested. So thank you.
Questions?
So with this clustering, can you hypothesize or tell us a little bit how different can the MHCs be that the TCRs are clustering and interacting with? So would you expect all those the things they're interacting with to be the same, giving the same peptide, or could they be varied?
So let me see if I understand the question. You ask if heterogeneity of the peptides contribute to the clustering that we see. So in this essay, I need to emphasize we don't use peptides and MHCs. We bypass the system using stimulating anti-CD3 epsilon antibodies. But people do conduct experiments with phosphorylated, peptide MHCs, like migdastin. And basically, we don't really know. This is the answer. I don't think that people have done these experiments, and this is really interesting to see.
In another in another presentation where you presented this work, you mentioned doing estimating the fractal dimension of the paths using box counting. Could you tell us a little about what you were finding out using that?
Oh, okay. So I didn't have time to go through all of our data, and there's much more imaging behind what I've shown. But basically, often what we see are fractals of point patterns, and these fractals are indicating some kind of a reaction, diffusion reaction of the proteins. Maybe proteins diffuse and then interact. I showed the cartoon of that. And then if you start, so the fractal dimension that we saw for LAT, for instance, is not indicative of DLA, diffusion limited aggregation, but rather of clustering of clusters. We get a fractal dimension of 1.4. So this is indicative of this kind of a process, and this is reinforcing our notion that there's some basal clustering of these receptors and these proteins, and then they coalesce into larger clusters. And we also know why, but this is a subject for another talk.
Okay. So I think in the first part of your talk you were using Jurkat cells. Is that true in the
Correct.
second part as well?
Yes.
So it's my understanding that Jurkat cells are lacking in P10, which is some people would know is the major phosphatase for PI3. So, and PI3 plays a huge role in terms of determining, and also the PIP2 that is the product, in determining membrane cytoskeleton and the way everything moves around.
Well.
So, do you think that could be playing affecting anything about the nature of the data that you're seeing in terms of whether it represents what's going on in a real T cell?
Okay, so I would say that Jurkat cells have been used for maybe a couple of decades as a model, as a robust model for early T cell signaling. Okay, and basically it's a system that has its deficiencies. Obviously, these are not real cells. We don't really know what is the peptide that the TCRs recognize, and some of the signaling might be deficient downstream of the TCR. Still, we know that they robustly flux calcium. So you have so PLC gamma, the PLC gamma pathway leading to calcium influx is intact, and we know that the phosphatases are recruited. CD45 is intact. LCK is at 70. So all of this has been controlled for. So and there are assays that show that the pathways that are activated in primary cells are the same as here. Maybe the extent of the activation is different, but
Well,
we're looking
you're
for the
missing the major
mechanisms.
negative regulator. I mean, I know a
There are
lot
many
of
negative
people who
regulators.
have played golf for like many decades, and there's
Well,
not very good.
so
even
the-
so I mean, just because people use Jurkat cells for a long time, I mean, now that we know
No.
that they're missing P10, and that's the major negative regulator that it affects all inositol phosphatase phosphate signaling, which plays a role in PLC gamma, as you know.
Fine.
Well, anyway, I mean, it would concern me.
Sure, but so again, I'm trying to limit what I'm saying to early T cell activation. P10, I would guess, is downstream of much of the complexes that I've shown. Yes, maybe there are differences, but my understanding is that the mechanisms, and people have shown these also in primary cells. Yes, you have the clustering. Yes, you have so people don't really know if there's cooperativity. But
Yeah, I mean,
we
the
think
fundamental
we do.
things that you've shown here,
Yes.
I know they've been seen in, I mean, it's all been seen in primary cells. I mean, what you're
Not all.
adding
Not all.
is this, well, what you're adding is the super resolution aspect, or some of
Well-
it, and some of the micro clustering. But all I'm saying is either add P10 back or try to move it to primary cells. I think that would be a good, you certainly could try to add P10 back.
Yes.
Anyway, I don't think
that's
it's
fair.
a detail. I think
That's
it's
fair.
not a detail.
Okay, so maybe we would disagree, but I'm saying something different. The message of the talk is that it's not seeing again these microclusters. These are nanoclusters. And you would lose basically diffraction limited imaging, average out most of the details that I've shown. So you wouldn't see the cooperativity in clusters, you wouldn't see the heterogeneity in the cluster sizes, etc. No, that's my.
You've added the super resolution
Yeah.
aspect to see that. We understand that. I'm just saying, do it in as realistic a system as you can.
Okay, it's not easy, and we can talk about that.
All right, let's thank everybody. We're going to take a couple of minutes.